So, as I said, we're going to go through some different things that are problems with small populations. So, the first topic is genetics. So, genetic diversity of a population is measured in a couple different ways. One is allelic diversity, which is the number of alleles present for a different gene. So, if you remember, an allele is a copy of a gene. So, it's the number of different types of a certain gene. So, if there was, for example, only one gene for eye color, you might have one gene that codes for blue, one gene that codes for green, one gene that codes for brown. Um, uh, there could be some other ones as well. But so you would count up the number of those each different versions of the gene, and that would tell you the number of different alleles there were for a particular gene or the allelic diversity. Heterozygosity is the percent of individuals in the population that have more than one allele at a gene locus. So if you remember, you inherit one set of chromosomes from your father, one set from your mother, so you have two copies of every gene. We're looking at the percentage of individuals that have two different copies. So one copy from dad is different than the copy from mom, for example. And so that would mean that you're heterozygous. So looking at heterozygosity is another way to look at this. Genetic variation is required for populations to evolve. Natural selection cannot occur if there is not variability in a gene. If all individuals have the same two alleles for a particular gene, then there's no variability for natural selection to act on. So genetic variation is really important. We want species to be able to evolve and adapt as environmental changes occur, and so they need genetic variation to be able to do that. Small populations tend to lose this genetic variability. So this looks at extinction risk and genetic diversity. And so this looked at different populations that would extinct and ones that are extant. And if you remember, extant basically means not extinct. So the extant ones are still living. And what we see here is on the x-axis, the average number of heterozygous loci. So a loci is, or a locus, loci is the plural of locus. A locus is a location on a chromosome. And so they looked at the probability of extinction here and the average number of heterozygous loci. And you can see here that there was about a 0.02 probability, so less than 2% probability of extinction here for certain numbers of heterozygous loci. And we see that populations that had lower amounts of heterozygosity had higher probability of extinction, where you got down here where they had higher numbers of heterozygous loci, you get lower extinction probabilities. We see that smaller populations lose genetic diversity over time through two different mechanisms. So this NE is effective population size, and we see that when that is very high, so at 1,000, if we go through multiple different generations, if you have that many, you don't lose any genetic variation. So this shows the amount of genetic variation remaining. So 1,000 loses no genetic variability. If you have 100 breeding individuals, you might lose 5%, but not very much over 10 generations. 50 individuals loses a little bit more, but you start to really get in trouble when you get 10 or less breeding individuals. This is breeding individuals, not all the individuals. And so once you get to there, over time, you lose more and more genetic variability. And if you get to really dire situations where there are only a handful of individuals breeding, you can lose genetic diversity really fast. So you can see it only took 5 generations to lose almost all of the genetic variability. And you'll also note that they start lower. So if you have less individuals, the amount of genetic variability you can have at a maximum is lower, just because there are fewer individuals to hold that genetic diversity. So 2 mechanisms, genetic drift and inbreeding. Genetic drift is evolutionary change due to chance. So we see there are multiple mechanisms for evolution. The ones people talk about the most is natural selection, where there are adaptations to environmental change occurring. Another way that evolution can occur is just randomly. So if a gene gets lost randomly, then that's technically evolution because it's change over time. That's all evolution really is. And so this is equivalent to sampling error or choosing too small of a sample size. In small populations, the outcomes can differ from theoretical expectations just due to chance. It's like you flip a coin a certain number of times. If you do it 1,000 times, you're almost always going to get about the same answer. If you flip it 4 times, you could get all heads. That's not improbable. And that's the basic idea with genetic drift. So it changes allele frequencies in a population from one generation to the next, but it does so randomly. It's not favoring any characteristic over another. It's purely by chance. If it's favoring one characteristic over another, then it's likely to be natural selection or sexual selection. If it occurs so randomly, it's more likely to be genetic drift. And so this is my coin toss example. You can see that if you increase the number of coin tosses, this graph shows down here, it's the number of heads you get. You would expect there to be 50% heads, 50% tails, right? So if you're down here, that means you got all tails, and if you're up here, you got all heads. So if you only flip the coin a couple of times, it's possible to get anything, all heads or all tails. But as you start tossing the coin, 40 times, 50 times, 100 times, 1,000 times, then you're going to converge on 50. And it's going to be very improbable that you get anything that's very different than 50% heads and 50% tails, assuming you have a fair coin. And so that's basically what this is illustrating. And it's the same thing that we're talking about with genetic drift. So if you have 1,000 individuals in a population, you're probably not going to lose much genetic diversity. The genetic diversity isn't going to change much over time. If you have 10 individuals breeding, you might get wildly different things just by pure chance of which genes happen to be passed on to the next generation. One type of genetic drift is a population bottlenecked. And this is when you have a large population that's very suddenly changed to a very small population. So it's a sharp reduction in population size due to some kind of random event. We most often think of this random event being some kind of natural disaster. So a flood wiped out most of the individuals, or there was a big fire and only a few individuals survived, or there was a volcano, those kind of ideas. But it could be human activities. If people found a new population of some really desirable species that they wanted to hunt, there might be also a bottleneck effect. But the survivors are basically a small random sample from the larger population. So this shows we have yellow and blue genes here in the original population. There's this bottleneck, so only a few individuals survive some catastrophe. By chance, most of them have this blue gene and only a couple have the yellow gene. And so those surviving individuals breed, and eventually the population recovers to be a large population again. But you can see now there's a lot more blue individuals with blue genes than yellow genes, and that's because of this bottleneck. So it often reduces genetic diversity, and we can see the signature of this in the genetics even in large populations. So it can have long-lasting genetic effects even if the population recovers. So here's another example where we've got lots of different colored genes. We've got purple and yellow and green. And in this catastrophic event, only the red and blue happened to survive this catastrophe. And so the yellow and orange and green and purple all are eliminated from the population. So you could see that the genes left after this bottleneck are very different than what we had before the bottleneck. So an example of this is the northern elephant seal. They were hunted almost to extinction. They got down to 20 individuals in the 1890s. And the population has recovered. There's now 30,000 individuals, but the genetic diversity is very, very low. And it's because those 30,000 individuals came from only 20. And so they lost a ton of genetic diversity in this bottleneck process. Another example is a founder effect, and it's kind of like a bottleneck, except there's only a few individuals that go somewhere to found a new population. So founder effect, they're the founders, kind of like founding fathers kind of idea. So there's this small colonizing population that have different frequencies of alleles than the population they came from. So only a small sample happened to start this new population. It's just the result of a sampling error. If different individuals had founded the population, you'd have different genetics in the new population. So an example is if a continental population of iguanas has 35 alleles at a particular locus, then the founding population of 15 lizards cannot contain all 35 alleles. And so these 15 lizards make it to a new island, and their genetics are very different than the one that's on the continent.